Well, thank you so much, Matt and Andrea, for inviting me to do a talk today for this webinar. I'll be discussing the management tools for Kona coffee root knot nematode. So I work for the USDA's Agriculture Research Service, the Daniel K. Inouye U.S. Pacific Basin Agricultural Research Center. And I just want to start off just telling you a little bit about our organization. So our mission is to conduct research for the development of sustainable agricultural systems and pest management programs in Hawaii, the Pacific Basin, and for all U.S. agriculture. And this is our facility located in Hilo, Hawaii. So the Agriculture Research Service is one of the world's premier research agencies for agriculture. And USDA-ARS scientists have done a lot for the industry. And some of the results of our research include higher yielding crops, increased disease resistance, new techniques for pest control, new cultivars with higher nutritional value, safer meat and poultry products, and also reduced production costs. We have a lot of student internships at PBARC, and I love this part of the job where I can get the youth interested in agricultural research so we can continue this tradition of helping the growers. So I just wanted to point out that the girl in the top right is Renee. She was one of my very first students. She did an internship when she was 16 years old. She then later went on to get her bachelor's degree and her master's degree. And now she's working as a permanent biological science technician here at PBARC in the genomics unit. And the young man on the bottom and now he works for the U.S. Border and Customs Patrol as an agriculture inspector. And that's his little inspector dog, Pogo. So I just wanted to share those success stories because that really makes me happy to see these kids getting jobs in the same field as us. So let's move on to nematodes. Today we're going to talk about plant parasitic nematodes, which are microscopic non-segmented roundworms. They have a stylet mouthpart for piercing plant cells, and even though they're tiny, they cause billions of dollars in damage to agricultural crops worldwide. Specifically, we'll be focused on the Kona coffee root knot nematode, Meloidogyne konaensis. It's a sedentary endoparasite, which means the female sets up a feeding site in the coffee root and manipulates the plant to continue to bring food to her so she can just focus on producing eggs, and a lot of them. She can produce hundreds of eggs in that egg mass, and you can see her body actually looks like a pearl. At this point, she no longer looks like the normal nematode worm. So it's a very interesting and complex system. This nematode is highly pathogenic on coffee. It completely destroys the root system and causes severe tree decline with mortality usually within 10 years. So how do these tiny little organisms damage crops so badly? Well, they destroy and weaken the root system because it's not just one, it's an army of them. There's thousands of them feeding on the roots of just one plant. So this interferes with the uptake of water and nutrients, which causes stunting of the plant and weakens the tree's immune system, resulting in poor crop yields. So what are the symptoms of the Kona coffee root knot nematode on coffee? Flagging and yellowing of the leaves is a common above ground symptom. And normally growers will think that their trees are lacking fertilizer or water. And yes, they are, but ultimately the reason is because, these worms have chewed up all of their roots. So they're unable to, uptake the nutrients that you're providing them. We see overbearing dieback quite often. Stump wobbliness is very common. I can go into a field and just usually when I look, I know already, they have a nematode problem. But if you can pull out one of your trees right out of the ground without any tools, you have a nematode problem. Um, so because a lot of the roots will be looking very quirky in the taproot, because this particular species of nematode will go straight into the taproot and destroy, your main part of the root system. So there's very few feeder roots. You'll also eventually get death and after decline of the plant. So this is a drone photo of a nematode infested field in late decline. The growers in the process right now of replanting with grafted nematode tolerant rootstocks. But nematodes usually kind of cluster. So you'll see specific areas that will look much worse than others. And also, if you notice in the top right of the photo, where there's like a low area, a lot of nematodes will accumulate there due to water runoff. So oftentimes you'll see very damaging areas in the low-lying areas. So what's this Kona coffee root knot nematode situation here in Hawaii? Well, it's very widespread in the Kona coffee growing region. I believe most of the farms probably are dealing with this problem. And unfortunately, it's becoming increasingly established in the ka'u growing area as well. We've done some recent surveys and we have not recovered it in Hilo, Hamakua, or Oahu. We still need to check Kauai and Maui. So as I mentioned, this particular species causes a severe decline of trees, resulting in a steady decrease in harvested yields per acre. So it's really a good idea if you as growers will keep track of your yields every year. That's, maybe one way to tell that, some problem is getting increasingly worse. So what are the management techniques that we can do about it? I'll be discussing 3 today: prevention, host plant resistance, and curative control. So prevention, this is my favorite one. Start clean, stay clean. It sounds simple, but honestly, this is where a lot of people mess up. You really need to propagate your plant material by seeds, cuttings, or tissue culture. Absolutely do not use pula-pulas. It's so easy to, pull up these seedlings out of the ground, and it seems very simple, but you're just starting your whole orchard on the wrong track by doing that, because nematodes love those little tender roots that grow on those seedlings. So think about doing everything in a nursery. And even a lot of times I've seen serious mistakes in the nursery stage. You want to keep your plants clean as long as possible. So please use sterile potting media, tools, and pots. Don't grow the plants on the ground. Put them on clean benches. Just think about good sanitation practices and be vigilant about that, and train workers in those practices. Once you get out to the field, it's really important to watch water movement from infested fields. Nematodes love to travel on water. That's another way that they get around, second only to infected seedlings. And never plant infested trees above areas of uninfested fields or areas that have previously been planted in coffee production. So next, we'll discuss host plant resistance, which I love because it doesn't require the growers to do anything different to their normal farming practices. Just start off with the right plant material. So we'll talk about grafting and traditional breeding. So grafting as a management tool is the most effective strategy for planting nematode-susceptible cultivars like Typica. So once you get nematodes into your field, eradication is impossible. So by using this route, you can easily adapt to the situation without having to put any additional inputs after planting. So there's 2 types of rootstocks: nematode resistant, which prevent root knot nematode reproduction, and nematode tolerant, which hold up in the presence of high populations of nematodes due to increased vigor. So we know for a fact nematode tolerant rootstocks increase coffee production. In a 13-year field study done at the CTARS Kainaleo Extension Station in Kona, we tested 8 rootstocks in an infested field trial. So the final year of the trial, the non-grafted Typica resulted in 81% mortality with less than 0.2 kilograms of red cherry per tree. While Typica grafted on Liberica, Meisner resulted in a 0% mortality. That means 100% survival and 10 kilograms of red cherry per tree. And in this photo, it's very obvious what the grafting can do to your orchard when you have a nematode problem. So without a doubt, nematode Liberica rootstocks increase coffee yields and tree health. So what is this Meisner I'm talking about? This is a new cultivar being released by CTAR. It originally was known, it's a coffee Liberica, originally known as Arnoldiana. But the name has been changed to Meisner in honor of Mr. Mark Meisner, who was the farm foreman of the Kona Research Station. And he dedicated 37 years of service to the university and to the farming community. And he helped maintain the collections. And he also helped many researchers do their research and get that information out to the growers. So you can go to the CTAR Free Pubs website and look up this extension bulletin, and it explains all about this new cultivar. And you can just see in this picture, the tree is very vigorous. And this is what makes it successful as a rootstock. The roots are just larger, more vigorous. The taproot is much larger, and this helps them to fight against the root knot nematodes. So please go to the website and check this out. And if you're interested in material, please contact Andrea and Matt. So let's move on to traditional breeding. That's typically when you evaluate nematode resistance in wild relatives, cross them with desirable commercial cultivars, and then my job is to screen the progeny. So I inoculate them with the root knot nematodes in a greenhouse setting. I allow that to reproduce for 1 year, and then I evaluate them for resistance and tolerance. How do we screen these coffee cultivars? Well, we have to wash the roots very carefully. We weigh them, we evaluate the root health. We do a root rot rating, check the galling index. Then we chop and blend up the roots to extract the nematodes. We sieve the solution, collect the nematodes, identify them, and count them under a microscope. So it's quite a time-consuming process, but that's what it takes to get the results accurately. So this is one of the trials that we did recently. We looked at Ethiopian Arabica, which came from the Hawaii Agriculture Research Center. Also a nematode-resistant Namaya cultivar. We looked at Obata and what we thought was Tupi, which I'll call Tupi Hawaii here, and we compared that to the susceptible Typica. And what we found was that the Ethiopian Arabica maintained its resistance even after multiple generations, along with this particular line of Namaya. The Obata did have some nematode reproduction. It was moderate, but still more than the other resistant varieties. And the Tupi was had very high levels of nematodes along with Typica. So that's how we evaluate for resistance. How we evaluate for tolerance is we have to take a look at the roots. So here we did a root rot rating, and we found out that Obata has a very low nematode tolerance. Even though it didn't have a lot of nematode reproduction, it's very, very susceptible to this nematode species, just as much, even more so than the Kona typica. So very scary to know that. So in conclusion, yes, they're good hosts. They're very susceptible. So unless you want your newly replanted field of CLR-resistant material to look like that picture, remember, continue to graft on nematode-tolerant rootstocks. So I'm really worried. A lot of the growers are so concerned right now about CLR, rightfully so. You really have to keep in mind if you're going to invest all that money and time into replanting, think about your roots. Because no matter how resistant it is against the rust, a lot of these new cultivars coming out are still very susceptible to our nematode. And if you just want to have the ultimate optimal conditions, just make that investment and graft. So let's discuss finally about curative control as a management tool. Um, lowering nematode populations in the field to reduce damage and increase yield. This can be done through cultural practices, amendments, and chemical and biological nematicides. You can apply this while the crop is growing, or you can treat the planting holes before replanting. So here's an example of one of the experiments we did looking at curative control. We wanted to take a look at chemical nematicides, especially the new ones that have been coming out, also biological nematicides, and some cultural practices. So we looked at fluopyram, spirotetramat, Burkholderia, some mycorrhizae, also looked at incorporating papaya seed and compost. And the results of that were in the Arabica plantings. Even though it wasn't statistically different, fluopyram did have lower numbers of nematodes than the water and the other treatments, and Burkholderia was a close second. So, like I said, this is looking at resistance, and this is looking at tolerance. We looked at plant growth, and that's really important because plant growth means more roots. It also means more production, more yield. So fluopyram definitely had the best plant growth, followed by the papaya seed amendment. So one thing about fluopyram is that I've seen that do this in many other crops as well. Somehow it doesn't always get rid of the nematodes, but it stimulates root production, and which ultimately stimulates plant growth and increases crop yields. And fortunately, the fluopyram has just gone through the IR4 program. And as Julie reported recently, it's just waiting to be put on the label by Bayer for coffee in Hawaii. So that's exciting news. I think it will be called Luna Privilege for the field applications. However, let's go back and look at these root systems. So you can see what Arabica does, what nematodes do, this nematode does to Arabica and then no treatment. You definitely don't want your plants looking like that. It's very damaging. In this study, the spirotetramat did not do well, but you can see the fluopyram increased that vigor in the root system. But wait, look at the last photo. So the Liberica, and this is the one we didn't even put any treatment on at all, but it still had the nematodes, was way bigger and way healthier. All the untreated Liberica were more vigorous than any of the treatments on Arabica. So really, do you want to put this kind of amendments and investment of applying this chemical when if you just start at the beginning and you put your investment at planting, you'll be much more successful. So my take-home message today for sure is just graft. And one follow-up on that is that healthy plants are much more tolerant to nematode infestation. With optimal growing conditions, plants are better equipped to fight off nematodes and other diseases. So make sure you always optimize fertilization and soil health, control insect pests and diseases as much as possible. If you need additional information, please go to the CTAR Free Pubs website. Besides the Meisner bulletin, there's also a pictorial guide to coffee grafting, as well as a root knot nematode sampling procedures manual. There's a lot of great information there. I would recommend going and taking a look. Just search CTAR Free Pubs and you'll be able to find it. One last note, I would like to introduce Landon Wong. He's a PhD student in nematology. He's working here at PBARC with us, and this is in collaboration with my graduate student mentor, Dr. Brent Sipes. So he's really going to do a deep dive into this nematode Meloidegain conensis. He's already started doing a lot of the morphology work. These are photos that he's taken with the microscope, and he'll be comparing our species of nematode with Meloidegain paranaensis in Brazil, which seems to have a lot of similarities in the destructiveness that it has on coffee. So he'll be doing also the genome sequencing and transcriptome sequencing, and helping me out with doing the transcriptome of the resistant coffee cultivars, so we can find molecular markers that can be used to speed up the screening in the coffee breeding program. So next year, we'll hear from Landon on the results that he's made. And finally, I would like to thank all my collaborators, especially Andrea and Matt and Nick, who helped me do all of this research that I've presented, as well as Kathy Mello, who is always there to help along the way to get this information out to the growers. So if you have any questions, please feel free to reach out. Thank you.